ಲೈವ್ ಹೋದ್ಯಾ? Hello, good evening. Hello, good evening, one and all. Okay. We will start. We will wait for 2 to 3 minutes. Let everyone join. Okay. When the views will be more than 50, then only I will start. So, फटाफट share कर दो सबको. Okay. We are discussing 2022 paper today. Again, very important. There will be so many questions which are new, which some are repeated. Okay. वेटिंग फॉर मैराथन क्लास अरे शुरू करेंगे भाई. थोड़ा बेसिक से जाएंगे ना, थोड़ा प्रीवियस क्वेश्चन पेपर देखेंगे पहले, उसके बाद एनालिसिस कर देंगे क्या आ रहा है, कहाँ से आ रहा है, उसके हिसाब से हम मैराथन में चलेंगे. ठीक है. Let me finish first the previous year question papers. There are some 3-4 classes. Then we will go for marathon. Okay, we will cover each and everything in a single go. Okay. शेयर कर दो फटाफट, सबको शेयर कर दो, सबको बता दो. There are only 16 viewers. सब मैच देख रहे हैं क्या? मैच तो खत्म हो गया. आ जाओ इधर, आ जाओ सब लोग. आरसीबी जीतने वाले है उधर. ठीक है, इधर आ जाओ, हम भी थोड़ा जीतना है ना जिंदगी में. अरे यार, मतलब कोई शेयर करने के लिए तो रेडी नहीं है, पता चल रहा है इसमें. आवाज सही से आ रहा है ना? इज माय वॉइस इज क्लियर? त्रिलोक बता रहे हैं सर, व्हाई डोंट आई स्टडी ड्यूरिंग एग्जाम्स. अरे भाई क्या कर रहा है? हाँ, पढ़ाई छोड़ के क्या कर रहा है? वही व्हाट यू आर डूइंग. If you are not reading, then what you are reading? What you are doing then? Clear? Okay. Okay, we are not going to sleeping. बस 40 दिन में नहीं है, 30 दिन बचा हुआ है. ठीक है, सब कुछ छोड़ दो, पढ़ाई, सॉरी नींद, ठीक है, खाना भी छोड़ दो थोड़ा थोड़ा. ठीक है, you just have to study for these 30 days. You have to do the revisions, okay, again and again, so that in your exam it will be very beneficial. Otherwise, definitely, okay, if you want to come in the single digit rank or 2-digit rank, so you should be very careful. You should do the revision more and more, so that you will get a questions in your exams. Okay, we are not going to waste a single minute from now on. Anyways, you guys already know about me. My name is Abhishek. I am from Karnataka. I completed my undergraduation from US Bangalore, and I got All India rank 21st in ICAR PG exam, All India rank 4th in ICAR PhD exam. I am currently pursuing my PhD in ICAR IARI, New Delhi. Okay, and I also cleared ASR net and joint CSIR UGC net. This is the first question of today's session. Okay, that is appear in 2022 exam. Which of the following geographical indication is not matched correctly? बताओ, आंसर बताओ, कमेंट में डाल दो जल्दी. सर कीप मोटिवेटिंग मी लाइक दिस, स्टॉप ईटिंग एंड ड्रिंकिंग. अरे भाई मैं बता दिया ना, इफ यू आर correctly utilizing these last 30 days, okay, then only you will get a good rank. Okay, it doesn't matter what you have done from past 2 years. Okay, these 30 days are very important. Okay, you just be with us always. Okay, there will be so many sessions are going on. Okay, there will be a pathology just now completed. Now the breeding is going on. Next genetics will come, microbiology. Okay, and seed technology. So you should guys be with us so that we will also give some motivation. Don't worry. बताओ answer बताओ. Kala Zira. Okay, this is not correctly matched. All other are correctly matched. Okay, the Kanda Aladi, it is from Odisha. Okay, very special Aladi. Darjeeling tea, which is the first JRF indication tag. Okay, the Darjeeling is district in West Bengal. Okay, and Tezpur Lychee, which is from Assam. Then what about Kala Zira? Okay, from where it is? Can anyone बताओ कुछ कोई? Anyone knows where this Kala Zira came from? From which? HP? No. It's not from HP. Rajasthan, HP, UK. Arey. This is Korapat Kala Zira rice. Okay, famously known as Korapat Kala Zira rice. Okay, you can see the grains will be black color. Okay, the what we can call the grain color, you can see there will be black color, but the rice grain will be somewhat brown. Okay, not completely black. You can see here it is a variety of non-basmati aromatic rice. I've already told regarding difference between aromatic and basmati rice. Okay, all basmati rice are aromatic, but not all aromatic rice are not basmati. Basmati having some special character, which is the grain length before cooking and after cooking. Okay, black paddy colored small rice mainly grown in the Indian state of Odisha. Okay, which is from Odisha. Your Kala Zira rice. Okay, it is from Odisha. They also got GI tag for this particular Kala Zira. Okay. Next question, match the following. Itna easy hai, theek hai? Regarding germplasm conservation, there will be one question. Okay, you can see how the questions are coming. You can easily expect the questions in the plant breeding section. There will be always one question from GI tag in last 3-4 year previous papers. You can see. Okay, there will be one question from GI. You should be analyzed like that. Okay, then only you can easily cover the syllabus also. You can see here there is one question from germplasm conservation. In last year paper also we have saw that there is one question regarding germplasm conservation. Okay, बताओ answer बताओ इसको. ये ठीक है. Germplasm conservation. Okay, there will be a in situ, ex situ, and cryopreservation. As I told yesterday, in situ conservation is nothing but in their particular habitat. Okay, the natural habitat, we are conserving the germplasms. Your national parks, okay, biosphere reserves, these are the in situ conservation. In ex situ conservation, we will conserve from out of their natural habitat. Okay, the gene banks, seed banks, okay, the different tissue culture technologies, cryopreservation, these are your in situ conservation. For A4, then clonal degeneration, that nothing but your virus infection. Okay, 1. DMSO is your cryoprotectant. 2. Virus elimination from cryotherapy. 3. 4, 1, 2, 3. 4, 1, 2, 3. This is your answer. Okay, what exactly the cryoprotectants we can see here. Again, there will be questions regarding cryoprotectant from past 2 to 3 years. Cryoprotectant are nothing but chemicals, okay, substances that are used to protect cells, tissues, embryos, and meristem, pollen, or seeds from the damage during freezing or cryopreservation. Okay, in cryopreservation, when we will store under 196 degrees Celsius, okay, this is the extremely cold temperature. Okay, when we are storing cells, tissues, or embryos or seeds, okay, the ice crystal will form in the tissues. Okay, there will be water content. Okay, सीड के अंदर टिश्यूज के अंदर लीफ के अंदर वाटर कंटेंट रहता है. ठीक है, व्हेन वी विल एक्सपोज दैट टिश्यूज टू -196 टेम्परेचर, okay, there will be ice crystal formation. Okay, what it will do? It will damage the tissues. It will do the some other effects for the tissues or seeds. So your cryoprotectants will avoid that ice crystal formation. Okay, you can see the functions of cryoprotectants. Prevents ice crystal formation. Okay, it reduces intracellular ice formation during freezing. Protects cell membrane, maintains membrane integrity during freezing and thawing. Okay, your cryoprotectants also maintains the cell membrane integrity. Reduce dehydration injury. Protect cell from excessive water loss. Okay, it will also reduces the dehydration. Improves survival after thawing. After thawing means when your tissues will comes to the normal temperature. After one, you will take it when you want to use, you will take it from -196 degrees Celsius. Okay, it will comes to normal room temperature. Okay, we will call that as a thawing. Okay, at that time, okay, it will improve the survival, your cryoprotectants. The normal cryoprotectants what we will use are DMSO, glycerol, sucrose. Okay, these are the different cryoprotectants, but your DMSO is very important. Okay, we will normally use DMSO in the most of the cases. ठीक है, बताओ, ये तो सिंपल है, शायद ये जनरल एग्रीकल्चर से आया है, मैं उधर उठा के इधर लेके आ गया. पोटैटो बिलोंग्स टू द फैमिली. छोड़ दो, इतना इजी तो एक दो क्वेश्चन आएगा. It will normally they will not come. Your composite is Asteraceae, your sunflower, safflower, Niger, Leguminaceae, your pulses, and Euphorbiaceae. Euphorbiaceae, there is a one good characteristics. The family, the species will comes under Euphorbiaceae, having latex in their tissues. Okay, for example, your rubber, castor, okay, cassava. Okay, these are the examples for Euphorbiaceae. इधर आ जाओ, next question. ICAR Indian Institute of Sugarcane Research is located in Coimbatore. Reason, ICAR National Bureau of Plant Genetic Resources is located in New Delhi. ठीक है, इस देखो क्वेश्चन देखो, अभी मैं बताया था ना कल, there will be a no relationship between both assertion and reasoning. Okay, sometimes they will give like this. It's actually a mistake of the paper setter. Okay, they can give it as a statement wise question, but now they are giving assertion and reasoning. Okay, there will be a different thing. बताओ अभी आंसर बताओ. D sugarcane research is in Lucknow. Yes Vijay. Okay, ICAR Indian Institute of Sugarcane Research, which is in Lucknow. Okay, what did he, then what is there in Coimbatore? ठीक है, sugarcane breeding institute. Okay, ICAR SBI, that is in the sugarcane breeding institute, which is in the Coimbatore. Your national NVPG is located in New Delhi. Then A is false, R is true. This is your correct answer. आ जाओ फिर से इंस्टीट्यूट्स. अभी देखो सब पेपर में फिर से इंस्टीट्यूट के बारे में पूछ रहा है. कल हम देखा था सियाट, IITA, नाइजीरिया, ओके, IRRI, CIMIT. Then now in this paper they have targeted our own institutes, okay, the national institutes, so that you should be aware of each and every institutes, okay, in on which crop they are working and the where they are exactly located. Okay, you should analyze like this. This will be the again पक्का क्वेश्चन. ठीक है, बताओ अभी बताओ, मैच करो इसको. सेंट्रल इनलैंड रिसर्च एग्रीकल्चर रिसर्च इंस्टीट्यूट, ये Your Central Inland Agriculture Institute, which is in Port Blair, okay. Your CREDA, that is Central Research Institute for Dryland Agriculture, which is in Hyderabad. Central Institute of Post Harvest Engineering and Technology, which is in Ludhiana. NIASM, okay, National Institute of Abiotic Stress Management, which is in Baramati. Indian Institute of Soybean Research, which is in Indore. Okay, A3, B5, A3, B5, C1. Okay, I ask your A will be your answer. अभी बताओ, कोई बताओ. The National Institute of Biotic Stress Management. Where is NIBSM? This is NIASM. Okay, National Institute of Biotic Stress Management. Where it is located? NIBSM. डाल दो, कमेंट में डाल दो फटाफट. NIBSM. अरे बताओ जल्दी. Which is located in NIBSM, which is in रायपुर. ओके, रायपुर छत्तीसगढ़. बरोंडा नहीं छत्तीसगढ़. हाँ वही है रायपुर. ठीक है, रायपुर छत्तीसगढ़. इधर आ जाओ. ये 2022 में दिया है. ठीक है, वो क्यों दिया है, ये तो उस समय कोरोना आया था. ठीक है, ये उसी को रिलेटेड है. अब देखो सही से देखो. Complete absence of false negative results would make a diagnostic kit highly successful. Complete absence of false negative. That means when you are having a coronavirus, okay, when they will test through the diagnostic kit, okay, if the patient is positive but the result is negative, okay, the result is coming like negative, that is your false negative. Okay, it is very dangerous. Okay, if we leave any one of the patient which is having corona positive to the public, okay, definitely it will spread. So complete absence of false negative, okay, of your diagnostic kit would make a diagnostic kit highly successful. Absolutely right. Okay, if you are getting complete absence of false negative, then it is highly efficient. False negative are due to poor sensitivity of the phenotypic or molecular marker deployed. Exactly correct. Why you are getting false negatives? Okay, the patient is having corona, but you are not able to find it, that positiveness. That is your poor sensitivity of the kit or markers. Okay, your reason is exactly explaining assertion. Okay, so your answer will be A. As I told yesterday, you can see here. There in assertion they are telling complete absence of false negative results would make a diagnostic kit highly successful. Okay, but then in reason they are not talking about false negative. They are talking about false negative are due to the poor sensitivity of the phenotypic and molecular marker deployed. Okay, there will be a not that much. We can tell that it's not exactly explaining assertion, but the paper setter is trying to make that one. Okay, he is trying to give a question like your reason is explaining assertion. At this kind of questions, okay, you should always choose the first option as I told. Okay, you don't think too much about it. Once again explain, sir. Complete absence of false negative is nothing but, okay, what exactly false negative? As I told, if you are having a corona positive, but your result is coming negative, that is the false negative. Okay, there is a positiveness, but the result is giving false negative. So that's why we will call it as a false negative. But if your diagnostic kit is having the good sensitivity, okay, and if very efficient, then there will be complete absence of those false negatives. If there is a positiveness, then it will show like a positive only. Okay, that's what exactly this assertion and reasoning is telling. Okay. Which of the following are two cultivated species of cotton used for spinnable fiber? बताओ स्पिनेबल फाइबर। आंसर लगा दो, कमेंट में लगा दो। डी डी डी। बताओ बताओ। Which of the following are two cultivated species of cotton used for spinnable fiber? Cultivated cotton. D D D. Yes. Okay. Your hirsutum and barbadans. We will also call this as a new world cotton. Okay. We will also call this as a new world cotton. What about Raimondi and Turberry? Those are the progenitors. Okay, the wild progenitors of your cotton. You can see your Gossypium arboreum, Gossypium herbaceum. These are Asiatic cotton. We will call them as a Asiatic cotton. Okay. Also, they are having diploid. They are having only one genome, A2, A1. You can see diploid in nature. For arboreum, we will also call as tree cotton or desi cotton. For herbaceum, we will call as a Levant cotton or desi cotton. These two are old world cotton. That means our Asiatic cotton. Gossypium, hirsutum and barbadans. Okay. AD1, that is tetraploid. We will call it as a upland cotton or American cotton, your hirsutum. Okay. And barbadans, that is also tetraploid. Okay. Egyptian or sea island cotton. We will call it as a sea island cotton. Your barbadans is Gossypium barbadans is having in fine quality cotton fiber. Okay. Fine quality cotton fiber. But your hirsutum is very high yielding. Okay. And also it is commercially we are growing in the larger area, hirsutum. But your barbadans is having fine quality of cotton fiber. Both are new world cotton. Okay. So your hirsutum and barbadans are cultivable spinnable cotton. But your Raimondi and Turberry, okay, again they are from old new world, but they are diploid in nature, and also they are like wild progenitors of your cotton evolution. They will come in the cotton evolution. So in maize, the opaque tube mutants are found to have some of the following quality parameters. Batao, opaque tube. Yeah, long staple. Exactly. Your barbadans. Long and fine staple cotton. C, high lysine, high tryptophan. Okay, this is mutant. Your normal opaque, okay, normal opaque 2 gene, okay, what it will do? It will produce gene protein, okay, gene protein, which is poor in your lysine and tryptophan. Okay, they are poor in your gene protein. But when it is mutated, okay, when it is got mutated, okay, that is your opaque 2, this is mutated version of your opaque gene. Okay, there will be a no gene protein, okay, immediately, okay, your lysine and tryptophan will increase. This is the basic thing what will happen in the maize kernels. Okay, when there is a normal opaque gene, there will be a gene protein. Gene protein is poor in lysine and tryptophan. But when that opaque 2 gene got mutated, okay, which will automatically increase the lysine and tryptophan content. Okay, this is the basic idea. Okay, there will be so many varieties. Okay, Vivek QPM9 and Shaktiman. These are the different varieties which are having opaque 2 gene. Okay. Yeah, these are the, again very simple and basic question. Okay, you can see. You can just see and just match the correct option. बताओ, comment में बताओ. लगा दो फटाफट. व्यूज तो बहुत कम आ रहा है. शेयर कर दो, सबको शेयर कर दो. बताओ, सबको बताओ. हाँ, इतना इंपॉर्टेंट नहीं है क्या प्लांट ब्रीडिंग? सी सी बी बी, अभी आ जाओ, इधर आ जाओ। हाइब्रिड, हाइब्रिड इज नथिंग बट फर्स्ट जनरेशन क्रॉप product has grown commercially. Okay, A is 3, only the 2 option are there. Pure line variety, pure line variety, the progeny of single cell fertilized homozygous plant. Okay, B is 4. Okay, B is 4. C, composite variety, not tested for GCA. Okay, the keyword C1, C1, this is your option. D for D, second one. Okay, regarding synthetics and composites, the procedure will be very same for the both the varieties. Okay, in case of synthetics, we will start, in the both the cases, we will start from the identification of superior inbred lines. But in case of synthetics, what we will do, first we will test those inbred lines for GCA, which are having good general combining ability. Okay, the superior combining ability, we will only choose those parents in synthetics. Okay, we will pool the seeds from mixture of seeds from those inbreds and we will grow in the open planted condition. Okay, there will be a intercross, possible intercrosses in the natural condition. Then we will mix those seeds and we will multiply the seeds and we will give to the farmers. But in case of composites, okay, the same procedure. We will start with the identification of superior inbreds. But at that particular time, okay, we will only see for the phenotypic performance. We will not test it for the general combining ability. Okay, the testing for combining general combining ability is nothing but we will cross with the OPV and we will see the progenies. But in case of composites, we will not do like that. We will just select some superior performance inbreds and we will mix the seeds of those superior inbreds and we will grow in the next season in the open planted condition. Then we will multiply the seeds and we will give to the farmers. Okay, this is the condition in case of composite variety. ठीक है, आपसे आंसर तो हो गया. Again, I just want to let you know, guys, that one who are joining new today, okay, there will be a price drop in our courses. Okay, we are getting at 1999 rupees till your exam. Okay, there will be 30 days for next year, and your agreed test series, that is for 799 for next year, and this year this is for 499. Okay, and the next year course you are getting at 4999. Okay, for 2027 exam, and for 2028 exam also we have launched the course which is you can get at 8999. Okay, you just see that there will be a complete coverage of syllabus. Okay, the one who are purchased already, they already know about it. Okay, you can just get some feedback from them and you can enroll to our courses. Which of the following constitutes genetic emasculation? लगा दो, कमेंट में लगा दो. Which of the following constitutes genetic emasculation? They are telling genetic. डाल दो फटाफट. B, ABC only, ABC. Your chemical hybridizing agent is an artificial emasculation. Okay, you are just using chemicals. Okay, कौन ABC? ABC. ठीक है. Your chemical hybridizing agents, for example, your Ibrex. Ibrex is the chemical which we will use in wheat crop. Okay, to make your plant as a sterile plants. Okay, they will kill the pollens so that you can do the crossing. Okay, this is the example for chemical hybridization agents. Okay, it is not completely genetic. All other genetic male sterility, transgenic male sterility, and cytoplasmic genetic male sterility, which is controlled by genes. Okay, in the crop plants. Okay, this is the simple question again. अरे आ जाओ, ये तो बार बार आ रहा है क्वेश्चन. देखो, you should see and you should know the anti-nutritional factors in the each crops. Okay, or phytochemicals which are present in each crop. Okay, your BOAA from Lathras, your uric acid from Brassica, Gossipal from cotton, linoleic from flaxseed. A3, B1 C4 लगा दो आंसर, ठीक है। In erythric acid, you already we are discussed about these kinds of things in the class very clearly about linoleic acid, linoleic acid, erythric acid. Okay, how it will convert erythric acid. Okay, what are the ways, what are the breeding technologies or breeding methods we can use to reduce that erythric acid. Okay, we have completely covered each and everything in our quality breeding class. Is it right? बताओ कोई हमारा स्टूडेंट्स कमेंट में लगा दो। मैं बताया था ना कुछ जीन, एरिक एसिड कम करने के लिए, हाँ, ओलिक एसिड इंप्रूव करने के लिए इन ग्राउंड नट, ठीक है। देयर विल बी जीन्स, ओके, FAD2, FED जीन्स, ओके, यू शुड नो अबाउट दोस काइंड्स ऑफ थिंग्स। ले लो ये टेबल, वेरी इंपॉर्टेंट, ओके, बार बार बता रहा हूँ, नेक्स्ट पेपर में भी ये क्वेश्चन में भी देखा था आज, ठीक है, इसलिए इधर ही बता रहा हूँ मैं, ये टेबल इंपॉर्टेंट है, ठीक है, पक्का एक क्वेश्चन आएगा। BOAA which is in Lathras, erythric acid in Brassica, glucosinolate in Brassica, again Gossipol in cotton, emagglutinin in broad bean, okay, phenolic glycoside in groundnut seed, cyanogen glycoside, okay, which is in forage legumes, phytic acid in pearl millet, trypsin inhibitor in soybean. Okay, there will be a definitely one question in cotton, Gossipol. Okay, there will be effect of that particular thing. The onion we consume is bulb, everyone knows, there is no confusion regarding that particular topic. Okay, you can see tuber, potato, swollen underground stem. Okay, it is nothing but your tuber or nothing but stem, underground stem. Corm, okay, Colocasia RB, gladiolus, okay, the gladiolus flower crop, okay, which is solid, swollen, again underground stem. Okay, your all are underground stem only. Okay, bulb, onion and garlic. This is again short stem with fleshy leaves. Okay, your bulb. And your rhizome, ginger and turmeric, banana, again horizontal underground stem. Okay, all are underground stems only. There will be different kinds of ways how they will store the food. Okay. The steps involved in the utilization of hybrid vigor in maize include. Okay. बता दो, आंसर बता दो. The steps involved in the utilization of hybrid vigor in maize. लगा दो, आंसर लगा दो. C. B, C, D only. Production of suitable inbred lines. Okay. Crossing of the selected inbred lines in the suitable combination to produce highly productive single cross hybrid, which is right. Okay. Mixing different single cross hybrid seeds to produce productive double cross hybrid. सब लोग बता रहा है आंसर B. मुझे पता नहीं कैसे वो B हो जाएगा आंसर. हाँ, B क्यों लगा रहा है? अभी आ गए सारे लोग ये. Crossing of the single cross hybrid in suitable combination to produce highly productive double cross hybrid. It is true. Okay, you should produce some single cross hybrid first. And production of suitable inbred lines, which is the first step in case of hybrid production. वो भी सही है, ठीक है. Mixing different single cross hybrid seed to produce double cross hybrid. ये सही है क्या? Just by using the single cross hybrid seeds, if you mix them, then will it make double cross hybrid? It is completely wrong. Okay, the answer is A, B, D. Answer is A. सब लोग डाल रहे हैं B और C. अरे क्या हो गया? Just think about it now. Okay, this is a simple question. If you make it wrong here, okay, this will make the difference between the good ranker and you guys. Okay, you should be very careful about these kinds of questions. It is very simple. They're telling mixing different single cross hybrids is to produce productive double cross hybrid. You can easily find out that thing. Okay, don't make mistakes. समझ गया? बताओ, कमेंट में बताओ. समझ गया ना ये क्वेश्चन? नूतन. ठीक है. Okay. The mating schemes that increases homozygosity in the plants. Which are the mating systems, okay, which will बताओ आंसर बताओ. B. A and C only. A and C. Okay. Your genetic assortative mating and phenotypic assortative mating. In case of dissortative mating, okay, it will, in case of genetic dissortative mating, obviously it will increase, okay, increase the heterozygosity. Your phenotypic disorder also balance or increase the heterozygosity. What exactly difference between genetic and phenotypic? Your genetic assortative mating is nothing but you are crossing the similar, okay, individuals which are from ancestry, okay, which are identical in nature genotypically. Okay, for example, sibmating, inbreeding, okay, between sister lines, okay, between the two inbreeds which are from the same parents, okay, that is your genetic assortative mating. What about phenotypic assortative mating? That is nothing but simply phenotypically similar parents. Okay, the phenotypically you are seeing the two inbred lines which are very similar and you are just mating among themselves. Okay, but in case of genetic assortative mating, it is similarity by ancestry. Okay, there will be some relationship between them. Okay, that is your genetic assortative mating. Okay, in case of inbreeding, okay, in development of inbreds, okay, you will follow the genetic assortative mating. Genetic dissortative mating in case of production of hybrids, you will follow the genetic dissortative mating. Okay, what about phenotypic assortative mating and phenotypic dissortative mating? Where we will use similar genome and similar phenotypic character, inter and intra species. वही है ना, genetic dissortative mating is nothing but your hybrids. Okay, when you are trying for hybrids. But in case of phenotypic dissortative mating, okay, as someone told, which is phenotypically very dissimilar, your distant hybridization, okay, your distant hybridization, nothing but intragenic, intraspecific crosses, okay, that kind we will use phenotypic dissortative mating. Phenotypic assortative mating in case of recurrent selection. Recurrent selection we will see now phenotypic, the superior phenotypic performance, then we will make intermate among themselves. Okay, that is your phenotypic assortative mating. Okay, these are the things you should know. Okay, we'll move to the next question. Inbreeding depression is the reduced survival and fertility of offspring of related species individual. Reason: inbreeding increases heterozygosity, exposing harmful recessive alleles in the homozygotes. Inbreeding increases the heterozygosity and which exposes the harmful recessive alleles in the homozygotes. Which is the correct answer? Inbreeding depression is an reduced survival and fertility. C. Yes. Your A is true. Inbreeding is nothing but reduced viability and fertility. Why it will happens? Okay, it will happens due to increases the homozygosity, not heterozygosity. It will increase the homozygosity and expose harmful recessive alleles in the homozygotes. Okay, in case of heterozygous condition, okay, in the cross-planted crop plants, okay, they will be in the heterozygous condition. Okay, your harmful allele, recessive allele, will mask from the dominant A allele. But when you will go for inbreeding, okay, when you will go for inbreeding, you will get capital A, capital A, small a, small a, small a. This will comes in the recessive condition, homozygous condition. So these kinds of plants will going to die. Okay, it will expose. Okay, your inbreeding will expose the recessive alleles in homozygous condition. Okay, this is the wrong statement. That is wrong reason. Okay, so your answer will be A is true, but R is false. The use of synthetic varieties. ठीक है, इस पेपर तो पूरा composite synthetics hybrids उस हिसाब से बना दिया. ठीक है, the use of synthetic varieties is more beneficial than single cross or double cross hybrids. The reason are why? If we assume that synthetic varieties are more beneficial than single and double cross hybrids, then what are the reasons? What are the reasons we can quote? B, C, D. Your first, yield of synthetic varieties are higher than hybrids, which is completely wrong. Okay, your yield of single cross hybrid is very high, then double cross hybrid, then your synthetics and composites. Okay, farmers can save their own produce as a seeds and no need to buy new seeds every year. That is right. Okay, in case of hybrids, okay, segregation will happen after F1s. Okay, so that you should always buy the new seeds every year in case of hybrids. But in case of synthetics, there is no need to purchase the seeds each and every year. Okay, synthetic varieties have wider adaptability than hybrids. Yes, exactly. Since the genetic base of synthetic varieties, what's the genetic basis of synthetic varieties? बताओ रिमी, Ankur, what is the genetic basis of synthetic varieties? The genetic base, we can say heterozygous, heterogeneous. Okay, yes, Vijay. Yes, so that those are wider adaptability than hybrids. Your hybrids are heterozygous and homogeneous. Okay, no need to make fresh crosses every year to produce seeds. Again, it is true. Your synthetics, there is no need to produce seeds. Sorry, no need to make fresh crosses every year. We will just leave it for open pollination for 3-4 years. Okay, so your B, C, D. These are the reasons so that we can tell the synthetic varieties are beneficial. But with respect to yield, you will always go for hybrids. Okay, if you want high yield and similarity, then you should always go for hybrids. In molecular marker technologies, the acronym of RFLP stands for. अरे हम ग्रेट स्टूडेंट्स को तो ये तो बहुत इजी हो गया, हम तो पूरा ऑपरेशन ही करवा दिया मार्कर्स का. बताओ. डी restriction fragment length polymorphism. Okay, which is in 1980 from Botstein. Okay, what about your RAPD marker? What is the full form of RAPD? बताओ. Full form of RAPD and who discovered that RAPD and which year? बताओ, कमेंट में डाल दो सब, फटाफट डाल दो RAPD के बारे में. RFLP is codominant marker. Yes, Shinch. Yes, RFLP is codominant marker. मैं तो clarity दिया है already, ठीक है, regarding different markers. Okay, which are dominant and which are codominant. You should always go for that. There is no need of confusion. ठीक है, RFLP is कोडॉमिनेंट लिख दो, मतलब सब किसी को पता नहीं है तो अभी लिख दो, ठीक है। ये लिख दो RFLP कोडॉमिनेंट, RAPD AFLP both are dominant. Your SSR SNP again codominant. These are the important markers. If they ask about caps, codominant. Scar, generally dominant, but we can also convert into codominant. ओके ले लो स्क्रीनशॉट ले लो फटाफट, ये तो फाइनल है, ठीक है। इफ दे आस्क एनी क्वेश्चन रिलेटेड टू डोमिनेंट एंड कोडोमिनेंट ऑफ डिफरेंट मार्कर्स, ओके यू शुड फॉलो दिस वन। AFLP RAPD dominant, RAPD dominant सही है। द फुल फॉर्म RAPD इज रैंडम एम्पलीफाइड पॉलीमॉर्फिक DNA, वो भी सही है। कौन दिया था, मैं पूछा था, who discovered RAPD, who given RAPD marker in which year, मैं वो क्वेश्चन पूछा था। आप लोग तो बस RAPD का फुल फॉर्म बता रहा है। कौन दिया था, अरे भाई बताओ जल्दी। इन 1990 विलियम्स, सही है ना। बॉस्टन, अरे बॉस्टन दिया था आपका RFLP, ठीक है, कंफ्यूजन मत हो जाना, ठीक है। Arrange the following in the order of their coming into existence. बताओ, इसको बताओ। The Indian Patent Act, International Convention of the Union or Protection of New Varieties of Plant Act, that is your UPAV. First one is 1961, that is your UPAV. 1970, that is your Indian Patent Act. Convention on Biological Diversity, that is 1993. Okay. Protection of Plant Rights and Farmers Right Act 2001, but came into force in 2003. Okay. Now you just arrange D, C, B, A. This is your correct answer. PPV, FRA. Note it, note down. Okay, PPFRA. Act is 2001. Force came into force 2003. Okay, authority 2005. Rules 2006. First variety registered in 2007. Okay, लिख दो, ये लिख दो PPFRA के बारे में। क्लियर, बताओ कमेंट में, बताओ क्लियर, बताओ जल्दी बताओ, क्लियर है ना PPFRA के बारे में सारे ईयर्स। लगा दो यार, लगा दो जल्दी। नूतन बस इमोजी डाल रहा है ठीक है। I think it is last question in this particular 2022 paper. Okay, they asked only 20 questions, very minimum question in this particular chapter. Again, as I told, there will be some coincidence with the seed tech and genetics. Okay, we can't differentiate them. Okay, whichever I left, those are from maybe floral biology. Okay, Prasad will take those classes. Means those they will cover those questions in that particular year. It is said that clone is an immortal. See. Exactly. Clone doesn't die. Clone can't be eliminated. These are not proper reasons. Okay, the performance of clone doesn't change with the time. The genotype of clone doesn't change over the time. Clones are nothing but your vegetatively propagated material. In case of vegetatively propagated crops, example onion, okay, your sugar cane, okay, when you are after generation, when you are using cuttings, okay, bulbs, as I told, stems in potato, okay, buds, okay, at that point of time, okay, your genotype of the clone will doesn't change over the time, so that we will tell that your clone is immortal. Okay, again, very simple question. Okay, yes, this is all about the 2022 paper, very easy, but where you should concentrate that the questions are coming from the same area, the same topics. Okay, there will be some 6-7 questions which are repeating, okay, year by year, but they are asking in the same area. For example, germplasm conservation, your IPR, PPBFRA, CBD, ITPG, you should know each and everything about this particular things. Okay, they are again asking about different hybrids, the different crosses, top cross, single cross, okay, heterobeltosis and standard heterosis. You can see, okay, you can easily get 100 marks. This is my motto. Okay, this is my statement. You can easily get 100 marks. Okay, by only analyzing the previous year question papers and our marathon classes. Okay, you can easily get. I will assure this point of time. At point of time. But one who are from our classes, one who are from our platform, a great platform. Okay, उनको तो सब पता है, वो मुझे पता है. Anti-nutritional factors. Yeah, Rimi, those are very important. We have already discussed in your classes also, I think. Yeah, I only took that quality breeding class, right? This is very important. Okay, yes. Multiline is homozygous and heterogeneous. Multiline is homozygous and heterogeneous. Exactly correct. Okay, at population level, it is heterogeneous, as I told you, as we discussed in the first class. And in case of individual level, that is homozygous. Okay, clear? Simple paper discussion. Okay, we'll see what they have asked in 2023 shift 1. There will be 2 papers in 2023, shift 1 and shift 2. Okay, we'll see those 2 papers in upcoming classes. Okay, there will be a 4 remaining paper. We will complete first these 4 papers and then we will move to marathon classes. ठीक है? चाहिए क्या marathon चाहिए नहीं चाहिए? और कौन चाहिए? मैं ले लो और कोई किसी को दे दो, बताओ. बताओ कमेंट में बताओ जल्दी. इंग्लिश, अरे भाई छोड़ दो यार, इंग्लिश हिंदी क्यों कंफ्यूजन हो रहा है. ठीक है, कॉन्सेप्ट तो समझ आना है. ठीक है, you will get that concept, don't worry. I will almost talk in the English only, but sometimes Hindi also come in between. Okay, बस पॉइंट्स आपको मतलब दिमाग को अंदर जाना है बस. I didn't understand your question, sir. I am asking, okay, I am asking, can I take marathon or should I give to someone else? Vijay Sant, Vijay Lee Sant. You sir, I didn't understand your question. Okay, yes sir, you only want it. Okay, need the app itself. अरे भाई ये क्या पूछ रहे हैं? मैम वेदर यू ओनली नूतन तो बस इमोजी में कंप्लीट कर रहा है चार्ट्स. ठीक है, गुड नाइट. ओके, वी विल मीट in tomorrow's class, which is 2023 shift 1 paper. Okay, till then, good night one and all. जाओ मैच. एंजॉय करो, मैच देखो बस।